Bismillahirrahmanirrahim. It's chapter number 6, nucleic acid and protein synthesis. First of all, what is a genetic code? The letters A, G, T, and C, they correspond to the nucleotides found in the DNA, and they are organized into the codons. And the collection of the codon is called the genetic code. For 20 amino acid, there should be 20 codons, and each codon should have 3 nucleotides to impart specificity to each of the amino acid for a specific codon. If we take one nucleotide, then it will show 4 combinations. If we take 2 nucleotides, it will show 16 combinations. If we take 3 nucleotides, it will show 64 combinations. That is the most suited for 20 amino acids. For example, you can see over there, this is the DNA, and this is the expression we are taking into our consideration. So this expression, it contains the nucleotide A, T, G, C, T, A, G, G, C on a template strand. So if we make the collection of 3 nucleotides, that is called the triplet A, T, and G, then C, T, A, then G, G, C. So we call it a codon. This is a codon. It consists of 3 bases, and if we take 3 nucleotides at a time, it is sufficient for coding the 20 amino acids, because in our body 20 amino acids are going to make more than 10,000 proteins. So for example, this triplet is responsible for coding this amino acid. This triplet is responsible for coding this amino acid, and this triplet is sufficient for coding this specific amino acid. These codes or codon are specific for each amino acid. So we are saying that this is the genetic code in terms of the codon, because collection of the codon is called the genetic code. Now, genetic code is the set of rules by which the information encoded in the genetic material is translated into the proteins by the living cells. Over there, translated mean after the process of transcription, protein synthesis. The information in DNA is in the form of the triplet codes, as we have talked about. That, for example, GCU is codon 1, ACG codon 2, and so on. So it is also found in DNA and in RNA. The information in the DNA is in the form of the triplet codons. It is first transcribed into the messenger RNA and then into the proteins. So every triplet codon in the DNA specifies one amino acid in the protein. So this codon message is shifted towards the from DNA towards the RNA, and later on protein is synthesized according to the information. Now, there are three types of RNA: messenger RNA, transfer RNA, and ribosomal RNA. Messenger RNA, it carries the genetic information of DNA for protein synthesis from nucleus to ribosome in the form of genetic code. This process takes place uh, inside the nucleus, that is called the transcription. Transfer RNA acts as the adapter molecule, carries the amino acid and drops it to the particular location by recognizing the codon on the messenger RNA by virtue of having anticodon. And what is the anticodon? We will discuss. Then the ribosomal RNA, it makes the complex with proteins and form the ribosomal subunits, which provide the space for protein synthesis. Single ribosomal RNA of the smaller subunit helps correct orientation of the messenger RNA during attachment with respect to the P and A side. We will discuss these two things in the translation. There are three types of RNA in a cell. This is the messenger RNA. Uh, as the uracil bases are present in the RNA instead of the thymine. This is the ribosomal RNA, the larger and smaller subunit consisting of the ribosomal RNA protein. This is the transfer RNA. It's a single standard structure which wrap itself in this manner, and at the other end it will pick the amino acid. So the transfer RNA will have the anticodon on its base. These anticodon will carry the complementary bases to the codon. So what is codon and what is anticodon? Codon is a sequence of three nitrogenous bases in DNA or messenger RNA, whereas anticodon is a sequence of three bases in transfer RNA, which is complementary to messenger RNA. Codon is complementary to a triplet of template strand, and the anticodon is complementary to a codon. The codon is present in the DNA or messenger RNA, whereas the anticodon is present in the transfer RNA. This is the structure of a typical transfer RNA. You can see over there, this is the transfer RNA. It has on its base the anticodon, UAC, and there is the codon AUG on the messenger RNA from direction 5 to 3 prime. So the codon and anticodon, they will have the complementary bases to each other. For example, A is following the facing the U to A, G to C. And that typical transfer RNA, according to its anticodon, which is again complementary to the codon, is carrying a typical amino acid on it, methionine. Later on, we will discuss about the process of translation, inshallah.